I'm Stephen Burley. I'm the director of the Institute for Quantitative Biomedicine, and I'm very pleased and honored to be able to welcome you here today to a joint presentation that will be given by myself and Professor Sagar Khare, who's also a member of the Institute and a member of the, like me, a member of the Chemistry and Chemical Biology Department, and also the Rutgers Cancer Institute of New Jersey. So we're going to be talking today about the protein data bank in the time of the coronavirus, and specifically about how the SARS-CoV-2 proteome has evolved during the first 6 months of the COVID-19 pandemic from the three-dimensional perspective. So I'll share my screen now and we'll get going. About halfway through, we'll change drivers and we'll present for the second half of the meeting, and then we'll take the questions together towards the end. So thank you again. So let me share my screen. And minor adjustment here. I'm going to go Is it now visible, Ken?
Yes, it is.
Wonderful. Okay. Thank you again for joining. We have a very robust turnout today, for which Sagar and I are very grateful. Our talk will begin introducing the PDB, explaining the role of the RCSB protein data bank in supporting global research and education, describing the impact of the protein data bank on drug discovery, and then outlining our efforts in the time of the COVID-19 pandemic. Sagar will then take the remainder of the time to describe unpublished work on SARS-CoV-2 protein evolution in 3 dimensions, with particular focus on on an antiviral drug target, the NSP14 Exoribonuclease N7-methyl G cap transferase, the methyltransferase. This is the first part of a 2-day affair. We'll have a description of the results of the project that we undertook over the summer with a big group of student summer interns, all of whom worked virtually. And then tomorrow we're going to do, with the help of colleagues who participated in the boot camp and also in the summer research experience for the dozen students, we'll do deep dives into NSP3, NSP5, the RNA-dependent RNA polymerase, the spike protein, and the pentameric ion channel E protein. And those of you who haven't yet registered for tomorrow's crash course starting at 1 p.m. Eastern Standard Time, please do so. That's the only way you'll be able to get the link. We're making the link available that way so we can actually get a handle on who's participating in the crash course. So thank you for attending today, and please attend tomorrow as your schedule permits. The Protein Data Bank was established as the first open access digital data resource in all of biology. We are days following entry into our 50th year of continuous operations, having been founded in 1971 with just 7 X-ray structures of proteins. Since inception, the archive has grown more than 24,000-fold to become the singular global resource for experimental 3D structures of biological macromolecules. It now holds more than 170,000 structures of proteins, nucleic acids, viruses, and macromolecular machines. Recognizing the global importance of the Protein Data Bank, which began operations here in the US, it's been managed since 2003 by the Worldwide Protein Data Bank Partnership, which includes our colleagues at PDB Europe, PDB Japan, and Biomag ResBank. The RCSB Protein Data Bank, the US-funded PDB, supports global research and education in fundamental biology, biomedicine, and the energy sciences. You see from this flow diagram here that we're integrating 3D structure data from all areas of science into the PDB archive and delivering knowledge through our websites to biologists, drug hunters, educators, students, and biopharmaceutical and biotechnology companies. As the PDB archive keeper within the Worldwide Protein Data Bank Partnership, the US RCSB Protein Data Bank has the special responsibility for safeguarding the archive, which has an estimated replacement cost of today approximately 17 billion US dollars. The US PDB, known as the RCSB PDB, was the founding member of the WWPDB partnership. We have teams at Rutgers, UCSD, and UCSF that support more than 40,000 depositors, meaning structural biologists worldwide. We deliver PDB data and related services to more than 4 million users around the world through our rcsb.org research-focused website that many of you have used. Our education and outreach website, pdb101.rcsb.org, served more than 600,000 educators and students in 2019. We're jointly funded and are grateful for it by the National Science Foundation, the US Department of Energy, and various NIH institutes. The graphic on the right shows the team as it was when we reached our 150,000 structure milestone, and we're now vectoring in on 200,000 structures. Colleagues and I recently published 2 papers analyzing in quantitative terms the impact of the Protein Data Bank on new drug approval by the US Food and Drug Administration. First, in a paper summarized in this slide, published last year in Structure, we showed that discovery and development of nearly 90% of the 210 new drugs approved by the US FDA 2010 through 2016 was facilitated by open access to PDB structure data. The impact of these structures ranged all the way from target identification and validation to structure-guided drug discovery, as exemplified by BRAF kinase and vemurafenib, shown on the lower right. This drug is used to treat late-stage melanoma. In a second paper published earlier this year in Drug Discovery Today, on the cover, we examined the impact of PDB data on new anti-cancer drugs approved by US FDA from the beginning of 2010 to the end of 2018. Discovery and development of approximately 90% of the 79 new anti-neoplastic agents approved by US FDA 2010 through 2018 was facilitated by open access to PDB data. We also showed that more than 70% of the 54 new small molecule anti-cancer drugs were the products of structure-guided drug discovery campaigns carried out by drug hunting teams in various companies in the biopharmaceutical space. In most of these cases, the company benefited from open access to structures of the target proteins previously contributed by academic structural biologists to the PDB. The Protein Data Bank is widely regarded as being comprehensive, authoritative, and of high scientific value. It has been certified by the Core Trust Seal as a trustworthy data repository and is regarded as a gold standard exemplar of open access to biological data as a public good. In this slide, I summarize the impact of PDB data on drug hunting. There are 5 stages in the process at which the PDB structures are playing a role. The first is knowledge of 3D structures informs our understanding of the protein's role in human health or disease and contributes to target validation. Second, close inspection of the target protein in 3D helps to identify potential drug binding sites, contributing to our assessment of the druggability of the target. Third, being able to visualize tool compounds binding to the protein helps us understand the chemotype binding properties of the target protein. And then quickly following on to structure-guided drug discovery, In all major pharmaceutical companies today, much of small molecule drug discovery is being driven by the opportunity to actually visualize how the small molecules in the lead series are binding to the target and to optimize those for both potency and selectivity. In the most favorable cases, there are also 3D structures of potential off targets. So the company drug hunters can also work to design out binding to a closely related protein that might represent an off target. It's this off target binding that is the source of most side effects of medication. And finally, 3D structures are also used during early stage drug development to optimize ADME properties. ADME stands for absorption, distribution, metabolism, and excretion. And the typical uses of 3D structure information in this area of the process includes avoiding inhibition of cytochrome P450s, avoiding binding to the human ether agogo channel, which can cause prolongation of QT intervals in electrocardiograms and potentially fatal arrhythmias. And finally, designing a way binding to the P-glycoprotein multi-drug resistant pumps is particularly important for anti-cancer agents. Now, turning to SARS-CoV-2, you know all too well, unfortunately, that in late 2019, an unexplained influenza-like illness was seen in Wuhan in central China. By the 10th of January 2020, we knew that the causative agent was a novel coronavirus dubbed SARS-CoV-2, and we knew the viral genome sequence. X-ray and 3D EM structures of viral proteins began pouring into the PDB since late January. The first of these was the structure of the NSP5 main protease, PDB-ID 6 Lima uniform 7, using the NATO alphabet. These structures and the portrait on the right, painted by our molecular artist, Professor David Goodsell, allow us to see the so-called unseen enemy. This portrait, eerily reminiscent of the opening of 2001: A Space Odyssey, shows the virus encapsulated within a respiratory drop. And this is the vector that is responsible for the bulk of transmission that we're seeing now throughout virtually every community in the United States. The US Center for Disease Control recently announced that everybody should wear a mask, practice spatial distancing, social distancing, and wash hands frequently. The reason for this is these aerosol droplets, these respiratory droplets that you see depicted in David's portrait. As of today, there are more than 500 SARS-CoV-2 protein structures freely available from the PDB, all downloadable with no limitations on usage. They're being used around the world to understand the biology of the virus and to discover new drugs and vaccines and to characterize neutralizing antibodies. The RCSB PDB has been hard at work responding to the pandemic since the first SARS-CoV-2 protein structures were deposited. We have prioritized deposition, validation, and biocuration of COVID-19 related proteins above all other structure depositions, and strongly encouraged immediate pre-publication release of these COVID-19 related structures. I'm pleased to report that nearly all of our depositors choose immediate release, as opposed to holding the structural information private until this scientific paper describing the structure is published. We've created a COVID-19 coronavirus resources page on our rcsb.org website that you can access easily. This is updated every week in response to release of new structures. We've also developed education and outreach resources, such as this hand washing video that you see in the animation, and many RCSB Protein Databank Molecule of the Month articles authored by David Goodsell, featuring the NSP5 main protease, the RNA-dependent RNA polymerase, and the spike protein. So at this point, I'm going to switch gears and talk briefly about the organization of the SARS-CoV-2 proteome and genome. and then give you a quick tour of some of the important PDB structures of SARS-CoV-2 proteins, and then I'll hand over to Sagar. The bottom of this slide depicts, the lower panel depicts schematically the organization of the SARS-CoV-2 genome and the viral proteome. Most of the non-structural proteins are expressed within two polyproteins named 1A and 1AB. And this these polyproteins need to be processed by two viral proteases. Both of these are non-structural proteins and are themselves part of the polyprotein. NSP3 PL proteinase that Sagar will be talking about in detail tomorrow cuts at the 3 sites denoted with the dark blue arrows. The NSP5 main protease cuts at 10 sites denoted with the light blue inverted triangles, and I'll be talking about NSP5 tomorrow. The structure of NSP5, the PDB-ID 6 Lima uniform 7, is depicted here. It's a homodimer. There are now more than 200 structures of this protein from SARS-CoV-2 in the PDB. Most of them have distinct ligands bound in the active site. In this pioneer inaugural structure, you can see a substrate analog covalently attached to the active site facing you from this half of the homodimer, and just peeking out from the backside of the other half of the homodimer, you can see the tip of the bound substrate analog. This is a drug discovery target with 2 active sites, and is the focus of a considerable amount of effort in both academia and industry today, looking for small molecules that will block the action of NSP5 and prevent the maturation of the polyprotein. Without the activity of this protein and the structure shown on the next slide, the virus would be unable to proceed with a productive infection, making the 2 proteases excellent targets for discovery. The so-called papain-like protease is part of a much larger multi-domain protein known as NSP3. There are both apo and co-crystal structures with inhibitors in the PDB, and one such structure is depicted here. This is a monomeric protein with 1 active site, and you can see the small molecule inhibitor sitting in the substrate binding groove here. This structure was the first of the papain-like proteases to come to the PDB, and the PDB ID is 6 Whiskey 9 Charlie. The RNA-dependent RNA polymerase is considerably more complicated than either of the 2 proteinases. It's a heterotetramer consisting of 1 copy of non-structural protein 7, 2 copies of NSP8, and 1 copy of NSP12. In this structure on the right, 6 Yankee Yankee Tango, you can see in light blue you can see NSP12. In the very dark blue, you can see the 2 copies of NSP8 that are involved in both protein-protein and protein-RNA interactions, and you can also see the single copy of NSP7 in this light blue-gray. And Tejaswi Venkatachalam will be presenting what we know about the structural biology of this system in the crash course tomorrow. This is the only validated drug target for SARS-CoV-2 at the present. It's the target of remdesivir, about which I'm sure you've heard a great deal in the news. Moving on to other potential drug targets, the NSP3 RNA helicase is responsible for unwinding the complex pattern of secondary structure that is found in the 5' end of the SARS-CoV-2 genome. And this is it's this RNA helicase activity that's responsible for the unwinding of the RNA and the processivity of the RNA-dependent RNA polymerase. There's now an EM structure of this protein, a drug discovery target, bound to the RNA-dependent RNA polymerase and a piece of RNA. And what the researchers at Rockefeller found, Liz Campbell and Seth Darst, is that 2 copies of the helicase operate with the RNA-dependent RNA polymerase to unwind the RNA. And you'll see more in detail from Tejaswi's talk tomorrow. Another important potential drug target is NSP14. This is the exoribonuclease and methyltransferase dual function enzyme that is responsible for both proofreading of the RNA and reducing the error rate, and also methylation of N7 of the 5' cap of the SARS-CoV-2 genome. One of the interesting features of the genome is that it's disguised by the virus to look exactly like a messenger RNA. It has a cap, it has a tail, and it's thought that NSP14 also works with the RNA-dependent and the RNA-dependent RNA polymerase and NSP13 to couple unwinding with RNA synthesis with proofreading during the process of genome replication. And the last potential drug target that I want to just give you a quick view of is the 2' methyltransferase that's responsible for the maturation of the cap. This is a heterodimer consisting of both NSP16 in light blue and NSP10 in dark blue. There are two potential binding sites here within the active sites, both the cap binding and the S-adenosyl methionine binding site. Both of those two sites represent potential targets for small molecule agents. Moving on now to some of the structural proteins of the virus. Many of you have heard about the spike protein. There are an enormous number of spike protein structures in the protein data bank, many of them determined by cryo-electron microscopy. And John Williams from the Baum lab will be talking in detail about this protein tomorrow. It's a target for vaccine discovery and neutralizing antibody discovery for passive immunization. And of course, the elucidation of this structure and complexes with the viral receptor on the surface of the host cell, angiotensin converting enzyme 2, have been critical for understanding fundamental virology. Equally important in terms of the structure of the virion is the nucleocapsid protein, which is responsible for RNA genome packaging within the envelope. It consists of an N-terminal RNA binding domain and a C-terminal multimerization domain. And it's this protein which is responsible for packaging the genome into a compacted RNA protein complex, which can the RNP, which can be enveloped by the virus, and embedded within that virus is the spike protein, of course, and at low copy number, an integral membrane protein known as the M protein, and then at slightly higher copy number than the M protein is the E protein ion channel. This is a pentameric ion channel, also potentially a drug target, and you'll be hearing from Grace Brannigan tomorrow about this protein and some of the work that was done during the summer, both by the summer researchers working with the PDB and the Institute for Quantitative Biomedicine, and also in Grace Brannigan's lab. So with that, it's time for me to stop sharing my screen to allow Sagar to do the same, and to thank you for your attention thus far. And I'm looking forward to Sagar's description of the as yet unpublished research project that we undertook this summer. Thank you.
Thank you, Stephen. I'm going to share my screen. Okay, so hopefully you can see a full screen view. If not, please let me know.
You're good.
Okay, thank you. So thank you, Stephen, again, for introducing all the proteins. And starting almost immediately after the pandemic broke out in the United States, Stephen and I had discussions about how the virus would be changing and whether we can understand it in 3 dimensions based on the structures that were, as Stephen said, pouring into the protein data bank. And so we decided to structure an activity involving visiting undergraduate researchers, as well as people from the RCSB PDB team and my lab, to work together to follow the virus as it is moving around the world and changing. And the way we did that was by obtaining sequences of the viral genomes that have been deposited in a central repository called GISAID. And we obtained all those sequences from GISAID and used those data to create models of all 29 SARS-CoV-2 proteins. So overall, we analyzed about 49,000 proteomes and identified so-called unique sequence variants for each of the 29 viral proteins. We classified them in terms of the chemical type, how many mutations there exist, and whether they are conservative, non-conservative, where in the protein they are occurring. And then we built models for all of these proteins, which were, we built about 10,000 models for all 29 proteins. So it was quite a substantial effort involving a lot of people, almost everyone in my group and a number of people from the PDB and a strong team of summer interns. So we built all these models, mapped them onto the 3-dimensional structure, analyzed them for their any functional perturbation that these mutations or amino acid substitutions might cause. And we also estimated the stability changes that are predicted to occur because of these amino acid substitutions using a software that my lab develops and contributes to called Rosetta, which was originally started in the lab of David Baker at the University of Washington. So we analyzed all of these USVs for changes in the stabilization free energy, which we called delta delta G apparent. And I'll tell you momentarily how we do that. So this is the team, and you can see that there is a lot of people here. And what we did was we first started with a boot camp in which we introduced SARS-CoV-2, and we focused our attention on performing the analysis of mutations on a particular protein of SARS-CoV-2, which is the COVID-19 main protease, or NSP5. And the students were trained to obtain sequences, construct sequence alignments and phylogenetic trees, and perform the visualization of the 3-dimensional structure, and also how to make mutations and estimate their energetic impact. About 12 of these bootcamp participants continued with us through the summer as part of the RISE undergraduate summer experience and performed the same kind of analysis on all the 28 other proteins. And these resources that we developed have been published in this paper and are also available on the rcsb.org website. Several people, I can see that many of you were instructors in the boot camp, and we thank you again for that service. And all of these lectures are available for everyone to see, and I certainly learned a lot during that course. So this is the picture of the summer interns. This is the RISE program, and I highly encourage you to advertise it to get really talented students over to Rutgers. And so most of these are college students, and except there is one very talented high school student who also participated. Okay, so let me tell you what we found, right? So we found first, qualitatively, that all 29 study proteins, all proteins of the virus, did undergo changes, did accumulate mutations. The mutations that we found were basically non-synonymous mutations. We did not find any insertions and deletions. And in terms of the mutations that were found, we found that most of the mutations were single mutations. In about 67% of the unique sequence variants had single mutations. A surprisingly large number, I would say, had double mutations, about 25%, and then a small number of triple, quadruple, and higher order mutations, including one USV which had 14-point mutations, which we think is probably a sequence artifact based on the fact that they are all consecutive. And as I said, no insertions and deletions. If you look at the identity of the mutations, what was the native amino acid and what that got substituted by, we found that mutations were conservative only in about 33% of the cases, whereas in 67% of the cases, these mutations were non-conservative, meaning a hydrophobic amino acid changed to, let's say, a positively charged, etc. So if we look more carefully at what these mutations are, here I'm showing you these what we call substitution matrices, where on the x-axis, you have the wild type or the native amino acid. This corresponds to the sequence of the virus that was first obtained in Wuhan, China, where the disease first broke out. And so we call that our reference sequence. And amino acids in that reference sequence are on the x-axis, and the substituted amino acids are on the y-axis. And these have been broken down according to where in the protein these amino acids exist. So this is for surface amino acids. So surface is defined as part of the three-dimensional structure that is accessible to water molecules. Core is, as the name suggests, the core, hydrophobic core of the protein, which is hidden away from solvent or water, and then boundary is between these two, and all of these mutations are combined into this matrix. And so a few things are interesting that I would like to point out. So the way these mutations, the amino acids are arranged is according to their chemical type. So first you have hydrophobics, then you have polars, then you have positively charged, negatively charged, and then cysteine, glycine, and proline, which are glycine and proline are special amino acids. So the first thing to notice is that hydrophobic amino acids undergo changes to other hydrophobic amino acids, as you might imagine, particularly in the core of the protein. And so do polar amino acids undergo changes to polar. But there are also changes like these that you can see. Alanine becomes D or E, which is aspartic acid or glutamic acid, which are negatively charged amino acids, right? So you have these hydrophobic amino acids becoming negatively charged. Normally, because I normally work in the field of protein design and protein engineering, whenever we're trying to modify a protein, the general rule of thumb is not to mess with glycine and proline, because these are special amino acids. They occupy very special regions of the Ramachandran plot, and other amino acids are not necessarily stable in these regions. However, you can see that both glycine and proline appear to be quite variable in the virus. And so we're assuming that all these viral sequences are, arise from viable viruses because they were obtained from patients. So clearly these proteins are functional, and therefore you can see that changing glycine or proline to other amino acids appear to be quite well tolerated. Okay, so what's going on with all these mutations, alanine going to D or E, or even vice versa, D or E going to alanine, right? So a lot of these mutations are actually arising because of the structure of the genetic code. So if you look at, let's consider alanine going to D or E, right? So if you consider this genetic code here, here is a wheel diagram of the genetic code where you have the first base in the center, the second base in the middle, and the third base, the wobble position on the outer spoke of this circle. So what you can see is that if you want to go from alanine to aspartate or glutamate, you really need to change only one central base, and you will easily be able to go from alanine to aspartate or glutamate, right? So, so many of the substitutions that we observe are occurring because of these type of single nucleotide changes. However, those are not the only changes that are observed. So for example, if you look at proline, going to aspartic acid, you will see that proline is here, on around 4 o'clock, on this genetic code diagram, and aspartate is on the other side. And so this would require 2 base changes, but we do still see that. So it's not just single base changes, but perhaps a large majority of them are occurring because of single nucleotide changes. So that's interesting to see that we can sort of rationalize where these things are coming from. Of course, we've made all these 10,000 models that we would love for other people to use as starting points for further analyses of individual proteins and as a proteome. So we'll be making them available publicly once the manuscript or a preprint is submitted to bioRxiv. Okay, so the other thing that I said was that we performed energy calculations. Let me tell you a little bit about those. So before I tell you about the results, let me tell you how we did the energy calculations. So we used, as I said, we used the Rosetta energy function to do that. And the way we estimate energy is by scoring a particular conformation of a protein. So essentially, the Rosetta energy function uses a combination of physically derived terms such as electrostatics, van der Waals interactions modeled by the Leonard-Jones potential, etc., along with statistical terms, which are derived from the protein data bank by considering the ensemble of structures in the protein data bank as a kind of thermodynamic ensemble. You can obtain sort of likelihood of certain configurations of amino acids being close to one another, or individual rotamers of individual amino acids. You can derive probabilities from this ensemble, and you can use that to score a particular conformation. So using this type of a hybrid energy function, we can use Rosetta to calculate the delta G apparent, or the Gibbs free energy of a particular protein model, which tells you the difference between the free energy of the folded form of which a crystal structure, let's say, is available, and the unfolded form. And then if we have a model of the mutant, then we can calculate a delta G, which is the change in the stabilization energy, by subtracting the delta G for the wild type from the delta G for the mutant. So we did that for all of these different USVs. And in order to generate the model for the mutant that we need to calculate the energy, what we do is we just basically perform a substitution. We just replace the side chain configuration by keeping the backbone fixed. Then we perform a Monte Carlo-based optimization of the side chain rotamers. And then we perform a gradient-based energy minimization of the entire protein to obtain a model of the mutant protein. Now, of course, whatever we do for the mutant, we also do, except for substituting the amino acid, we do also for the wild type in order to create our reference model, and then we can subtract these energies in order to obtain the delta G apparent. So what did we find when we did this analysis? We found that 72% of the mutations were destabilizing. So their calculated energy change was from 0 to 15 Rosetta energy units, and Rosetta energy units is like a kilocalorie per mole. So protein stability is typically between -5 to -15 kcal per mole. So that's why we chose a threshold of 15 REUs to define what was destabilizing. So you can see that most of the mutations were slightly destabilizing. About 24% of the mutations were actually stabilizing with an energy effect which was negative. So that caused the protein to be more stable. And as I'm sure many of you who might be aficionados of modeling and crystal structures will realize, doing 10,000 calculations and is going to yield some results that are unreliable. And in 4% of the cases, we obtained energy changes that are more than 15 REUs, perhaps indicating that our assumptions about keeping the backbone during our modeling fixed was being violated in these particular cases. And those might be interesting candidates to perhaps characterize structurally even more to understand what's going on. Okay, so let's look at the data that we obtained. If you plot a histogram of the energy values that you get, so here on the left, you can see that In our data, we can clearly see, it follows a skewed Gaussian distribution with a second peak or a fat tail on the positive side that corresponds to another Gaussian peak. So it's best fit by these two Gaussians with peak values corresponding, or the mean values of the two Gaussians are, plus 1.3 REU or Rosetta energy units, and the second one is at plus 6, roughly REU or Rosetta energy units. And so where are these two Gaussians coming from, right? And it turns out that, in fact, if you look at the distribution of stability effects from experimental determinations of delta G's, you can see that those data also correspond to similar bi-Gaussian distributions. So here, this is from the work of Nobu Tokuriki when he was a postdoc in Danny Taufik's lab at the Weizmann Institute. What Nobu found was that if you perform computational calculations on about 15 or 20 different proteins, and you plot a histogram of their delta G's, you would get a similar bi-Gaussian distribution. In this case, you're assuming all possible mutations at all possible sites in each of these proteins. And in fact, if you do it for some experimental measurements on a model protein like TEM beta lactamase that other people had done, Nobu found that those distributions in the experimental data set also corresponded very well with this bi-Gaussian distribution. You can see this fat tail at positive delta G values. In fact, Eugene Koonin then performed a similar kind of computational analysis on entire proteomes. So you can't read the legend here, but this is Saccharomyces cerevisiae, Homo sapiens, E. coli, and another thermophilic organism. So 5 proteomes, whatever proteins are available in the protein data bank, if you perform in silico all possible mutations at all possible sites and plot the histogram of delta G, you would similarly get this kind of bi-Gaussian substitution. So what we see is that the virus is essentially following these rules of protein thermodynamic stability, even though the number of mutations that have arisen in the virus, which are roughly 1 per site instead of these are 19 per site, are much lower. So that is nice to see. So where does the second Gaussian peak come from? And in order to explore that, we plotted the histogram of delta G's corresponding to core residues, surface residues, and boundary residues that are shown in blue, green, and orange respectively. And what you can see by comparing the mean values for the entire Gaussian with these 3 Gaussian distributions is that the first peak, which corresponds to plus 1.3 REU in the entire distribution, comes largely from the surface residues, whereas the second peak comes from a mixture of boundary as well as core residues. And in fact, in the Tokuriki paper as well, when they plotted the experimental data set from wet experiments, they were able to see that the core distribution, mutations in the core residues were responsible for the second peak as well. So what is heartening to see is that these distributions reflect, in some sense, in the SARS-CoV-2 mutations, reflect the universal constraints of the different microenvironments in a protein, which is, surface residues have less constraints, and so their impact is also lower, whereas core residues are obviously more constrained and therefore have a kind of broader distribution of energetic effects where they're shifted towards more destabilizing values and can lead to more stabilizing as well as destabilizing effects. And so this is heartening to see that that the mutations that have occurred so far follow that trend. In the remaining time, I will quickly give an introduction to the type of analysis that was done for NSP14, that, as Stephen mentioned, is an exoribonuclease and methyltransferase, has 2 activities, and it forms a heterodimer with NSP10 and has 2 active sites corresponding to its 2 activities. I'll just summarize what we found for NSP14, and in tomorrow's crash course, we'll be looking at other systems in more detail. This is just to give you a sort of taste of the type of things we'll be looking at. So as I said, and as Stephen said, it's a bifunctional enzyme. It has a methyl transferase, which generates a cap to hide the viral RNA, and it has an exoribonuclease for RNA proofreading. Now, it turns out that there is no crystal structure available for SARS-CoV-2 NSP14, but it is 95% identical to the SARS-CoV-1 or SARS-CoV NSP14. And so we built a homology model based on that. And then we looked at the viral proteomes and analyzed where these changes had occurred. So first of all, 83% had NSP14 unchanged. So, because this is a proofreading enzyme, it clearly shows you that it's doing its job, that most of the NSP14 variants that were found had an identical sequence, so proofreading was working. There were about 335 unique sequence variants, and we found that most of them were single. There were a few doubles, and there was one 14-point mutant that was all mutations occurred in sequence contiguous regions, a stretch of residues, which makes us think that it is most likely an artifact. As with other proteins, most of the substitutions are non-conservative, and also in terms of the distribution of energies, we also found that most of these mutations are moderately destabilizing and occur mostly in the boundary and surface, with only 8% in the core of the protein. If you look at the key active site residues for the methyl transferase activity, there are two things that are important. One is to bind the cap that is being added, and the second is to bind the cofactor or SAM, which is a key feature of most methyl transferases. And so you can look at the crystal structure or the homology model and identify residues that are involved in both cap binding as well as SAM binding. And while small changes were observed in both cases, you can see that these changes are largely conservative, D going to E, Q going to H. Basically, you can still make hydrogen bonds, and polars going to polars, hydrophobic F going to leucine. So the impact is been modest, which is a good thing, because it means that drugs that are developed to target this are likely to hit a large number of patients. If you look at now the exoribonuclease active site, there is a conserved dead H motif, which corresponds to a variety of negatively charged side chains, aspartates and glutamates, and a histidine 268. And in fact, for this particular active site, as one might expect, this activity is key for viral survivor, and none of the residues were changed in viral samples that were viable, presumably, and derived from patients. Because if there were changes, you can imagine that the error rate would become really high, and there would be a so-called error catastrophe leading to the virus dying out. So, so to summarize what we learned, first is that mutations are relatively rare, so proofreading is working. Only a small number are in the core, and many non-conservative changes are likely result from single base changes. So both the physical constraints on protein stability as well as the genetic code can explain the type of mutagenesis that has occurred. The distribution of stability effects follows a universal bi-Gaussian distribution, where core mutations, even though small in number, can correspond to fat tails on the positive side. And also that key enzyme active sites, for example, proteases, NSP14, the RNA-dependent RNA polymerase, represent attractive targets for structure-guided drug discovery because they are highly conserved and necessary for viral survival. Finally, the methodology that we developed, we're very excited that it can potentially be used for a variety of things going forward. So for example, for structure-based drug design, one can imagine that there are 200 structures of fragments bound to the main protease available, we can make mutation models of substitutions and perhaps find substitutions which may inhibit or decrease inhibitor binding without having any effect on substrate binding. And these would be candidates for drug-resistant mutations, for antibody-bound crystal structures or cryo-EM structures of, let's say, the receptor binding domain. One can look at and see if there are likely to be any escape mutations, and one can sort of prospectively identify them and hopefully help deal with those things better. So let me just end by thanking the team from my lab that did this work. So Joey Lubin, who is in the audience I just saw, really spearheaded the development of tools that we have used, and he was developed the bootcamp materials. And after Joey left us to go for a short stint at Merck for an internship, Changpeng, Zhofan, Elliot, and Vidur and Lingjun, all of them worked together. We really enjoyed working together in the summer to deal with all kinds of proteins. We had, for people who do molecular modeling, we had everything from membrane proteins to oligomers to heteroligomers and all the attendant quirks that I'm sure you can appreciate that are associated with modeling such a diverse set. The team dealt with in a very excellent manner. And of course, thanks to the PDB team, bootcamp participants, and RISE summer interns, and Virginia Jiang from Scott Banter's lab at Columbia, who helped us with some membrane proteins, and John Williams and Cody Hoop, who helped us with the spike protein. And with that, I'll pass it back to Stephen.
Thank you very much, Sagar, and I thank you all for your attention. I'm only able to be here today because of the huge team that stands behind me at the RCSB PDB. There are pictures of all of the current team members here, plus our director emerita, Helen Berman, known to many of you. And I want to acknowledge again the funding we received from NSF, NIH, and the US Department of Energy. Thank our hosts here at Rutgers, UC San Diego, and UCSF, and acknowledge the importance of the Worldwide Protein Data Bank partnership. And then maybe, is there one more slide, Sagar?
Yeah.
Yes, I just wanted to provide another plug for tomorrow's crash course that Sagar and I are co-chairing. So after opening remarks from the Executive Vice President for Academic Affairs, Professor Prabhash Moge, David Goodsell will talk about all of the viral proteins and their role in the life cycle from his perspective as a molecular artist. And then Sagar and I will talk about the proteases. Tejasvi will talk about RNA-dependent RNA polymerase. John Williams will describe the structural biology and the mutagenesis studies of the spike protein, and Grace Brannigan will do the same for the E protein. So it'll be a very, very intense afternoon, and I hope that all of you and others who've registered online will be able to join us tomorrow. If you want the Zoom link, you must register. Please use the link shown on the slide here. And with that, we have a few minutes for questions. So I'll open up the chat box if people want to post questions in the chat. So there's a question for you, Sagar. I saw there's no experimental structure of a SARS-CoV-2 membrane protein. How did you validate the same for your work? How did you determine the oligomeric forms, if any, for this paper?
So we did that depending on the protein. For the membrane proteins, I think we mostly built homology models based on the SARS homologs. Most of the membrane proteins, I think, are highly similar to the SARS homologs. And,
in fact, all of the proteins are highly similar to the SARS homologs.
Yeah. Exactly.
The key difference between the two viruses is one's, they're both bad, but one is infectious in the asymptomatic stage and one is not. And that's why our first brush with a major coronavirus epidemic across the species barrier in 2003 wasn't that bad. Only 800 plus people died worldwide, no one in the US.
Yeah. And then in terms of oligomeric states, yeah, so either that was based on the biological unit in the PDB in the case of proteins where we knew or had a crystal structure, or it was based on looking at the literature. And sometimes it took us a little while to decide what the oligomeric state was, so that was a key consideration. And sometimes we chose a functionally more relevant form. For example, I think NSP13, Stephen can correct me if I'm wrong, where it appeared based on the crystallographic structure that it might be a dimer, but looking at the functionally relevant form, it was monomer, and so we used the monomeric form then.
That assertion that Sagar just made was borne out by the work from Rockefeller that showed the helicase in action in tandem with the RDRP. Monica Roth is asking, I thought the N protein was a 2-metal active site, and people were suggesting that integrase-like inhibitors might be good leads. Our structural knowledge of the N protein, the nucleocapsid protein, is quite limited. It's just limited to 2 domains. And so it may be that we simply weren't able to consider that, Monica. We will check before we submit the paper. The endonuclease. Yes, the endonuclease has a magnesium ion in the active site, Monica, and we have a structure of the SARS-CoV endonuclease, but not the SARS-CoV-2. Ricky Cohen is asking, being asymptomatic but contagious means that our immune system is being super suppressed. And then he goes on to ask a question about ORF3b and its role in modulating the impact of interferon. So Ricky, and for everybody's benefit, I don't think that that being asymptomatic but contagious means that the immune system is necessarily suppressed. It's just that before people begin showing symptoms, they are spreading the virus, and that didn't seem to be the case for SARS-CoV in the early noughts. You're right in identifying ORF3b as one of a number of proteins encoded by the viral genome that modulate the host immune system. Sagar, perhaps you want to comment on, just give a brief preview of the role that the PL-Pro plays in that regard.
Yeah, so the PL-Pro, it has peptide hydrolysis activity, and it not only cleaves the viral polyprotein in the 2 or 3 places that were indicated before, but also cleaves off ubiquitin and ISG15, ISG15 is a ubiquitin-like protein, which is a mark from host proteins. And so in that way, it interferes with some immune responses. And so the virus not only kind of hides its RNA, for example, but it also actively cleaves the host proteins in key locations to evade detection and immune response.
And the other thing it does is to degrade the host mRNAs. That's another. There's another.
Right.
There's an exonuclease which is responsible for degrading some of the host mRNAs. And so that's yet another mechanism through which the virus is able to elude the immune system. Antonio Meroli asked, do you think the spike protein can be cleaved at the base of its extramembranous stalk or just pops out? I don't know the answer to that question, Antonio, but I'm hoping that tomorrow John Williams will comment on that when he presents. Stephen Zhang asks, how related is NSP14 to the human CAP methyltransferase? We haven't looked at that, Stephen, and we need to do so. Good point, thank you. And then a question for Sagar from Shishir Chundawat. Can you comment on some critical mutations that were seen on the spike protein and D614G is of course cited and and its receptor domain. And again, I would suggest we defer these questions to John tomorrow.
Yeah, I agree. I
Since
think
he'll be giving a full exposition on what we learned about the sequence variation in the spike protein.
Yeah, and it's really very interesting. I don't think that exists a very clear answer, but yeah, you'll find out tomorrow from John.